Thank you very much. I am Moimedi from University of Nagasaki Institute of Tropical Medicine. I'm going to talk about the current situation and challenges in the development of dengue fever virus. 1/3 of the global population live in dengue fever endemic regions, and the annual number of patients is 400 million. This virus is mediated by mosquitoes, and the endemic regions include mostly areas where many people live, and the infectious route is between humans and mosquitoes. In Japan, there were outbreaks of dengue fever in the past. A major outbreak was repeated between 1942 to 45 in Kobe, Osaka, and in Nagasaki. 200,000 people were infected. The vectors are Aedes aegypti and Aedes albopictus. Aedes aegypti live in areas south of Tohoku. This mosquito is active in summer and in fall, and that is where there's a heightened risk of endemic. What this shows is the global warming and expanded mosquito-mediated infections. A shows the areas of high risk of mosquito-mediated infection in the 1990s. However, after 100 years, with global warming, high-risk regions are to be expanded. In other words, the risk of mosquito-mediated infection is also expanded together with the global warming. What this shows is the number of patients of dengue fever in Japan. In the 1990s, the number of patients is around 10. But starting from year 2000, we see an increase of annual number of patients. And in year 2010 and onwards, there are several imported cases reported every year. Furthermore, in 2014, there was a domestic outbreak in Japan. The lower panel shows the fact that most of the imported cases relate to the travelers returning from Asia. What this shows is the dengue fever outbreak in 2014 in Japan. The first case was reported in August, and the outbreak was prevalent between summer and fall. If you look at the gene sequence of the virus, this was the serotype 1, genotype 1 virus, which was similar to the mosquitoes found and virus found in Southeast Asia. So it is suggested that the virus came from these regions. Major symptoms of dengue fever and hemorrhagic fever include, in case of dengue fever, headache, pain of joints and muscle, and thrombocytopenia. Some patients are asymptomatic or symptom-free, but about 30% of the patients exhibit these symptoms. Among dengue fever, there are severe cases leading to dengue hemorrhagic fever. Symptoms of dengue hemorrhagic fever include more tendency for bleeding and plasma leakage, DIC, and there are some fatal cases. This shows the dengue fever ICU in the south of Vietnam. This is photo from Children's Hospital. As you can see, during the endemic period, we ran into shortage of beds, and there are many severe cases. So this is a very serious case of public health, as there are more than 100,000 patients per annum in the endemic regions. This shows the clinical course of dengue fever. There are 3 phases: febrile phase, the critical phase, and recovery phase. In the critical phase, shock related to bleeding and multi-organ dysfunction may occur. After 24 to 48 hours of critical phase, you will have a recovery phase. But severe dengue cases can be classified into 4 grades. The gravest case, grade 4, or in case of DSS, the mortality is around 25%. The mechanism of onset of dengue is explained with several different theories. Generally speaking, for primary infection, virus plays an important role. In case of secondary infection, infection-enhancing antibodies, T cells, and host factors play important role, host factors being genetic background of the host. In case of dengue fever, the mechanism for the development of severe case is explained with the antibody-dependent infection enhancement. According to this theory, one person is infected by a single serotype, and when that happens, this person will have a permanent defense mechanism. But if the person is reinfected by different serotype, like type 2, if there's no defense mechanism for this, then cross-reacting antibody and the virus will form a complex, leading to an infection enhancement, and this will exhibit more severe symptoms. Dengue virus has 4 serotypes, but all of these 4 serotypes exhibit and develop dengue fever and hemorrhagic fever with similar symptoms. In the primary infection, antibodies are induced, but these antibodies are not necessarily effective as a defense for other types of virus. During the history of dengue vaccine development, 70 years ago, when the virus was first isolated, the development of vaccine began. First, the virus, which is live, is used to inoculate mouse to develop mouse-derived vaccine. But this vaccine was not attenuated. So, using this vaccine, cell-cultured attenuated tetravalent vaccine was developed. Live virus is used for dog kidney cells, marrow cells, and other cells for attenuation. But again, attenuation was not successful. So the study was terminated during phase 2 clinical study, and came the development of a recombinant chimeric virus, which is tetravalent vaccine. The development started in the 1990s. Yellow fever virus serves as the backbone for this vaccine, and attenuated dengue virus was used as a backbone, or gene deficient virus was also developed. On the other hand, more recently, inactivated virus, subunit, DNA vaccine, and viral vectors were under development. There are challenges for the dengue vaccine development. During the development, the most important thing is to confirm the safety and efficacy of the vaccine. However, for dengue fever, the mechanism of infection-related defense is not clear, and the neutralization and infection enhancement, how these two conflicting phenomena have interaction, and how can we induce sustained defense. These are very important things about the development of vaccine. In 2013, the first tetravalent vaccine was studied. Among the subjects with the neutralizing antibodies, there were many dengue fever developed. And in 2017, subjects with inoculation developed a dengue severe cases, and there were some fatal cases. So whether or not the neutralizing antibody, which is an important marker, can reflect clearly the level of defense is a question. These are dengue vaccines under development. Recently, various types of vaccines are under development. They are in different phases of clinical development, among phase 1 to 3. But the Sanofi vaccine I just referred to earlier was withdrawn in the Philippines. This was because of the fact that there were many severe cases among the inoculated subjects. This led to the withdrawal of the vaccine. Why is it that it took 70 years and still we are not seeing the successful vaccine? It is because the mechanism of defense is not very clear. Generally speaking, for primary infection of dengue fever, virus infects the target cell, and dengue fever symptoms are exhibited, and these symptoms are mostly mild. In case of secondary infection with the same serotype, the neutralizing antibody from the primary infection serve to protect the infection, and this leads to the defense mechanism. However, with the secondary infection with the different serotype, neutralizing antibody from the primary infection may not serve as a defense. So this antibody and the viral serotype will form a complex, and this complex through Fc gamma receptor cell lead to the production and replication of many particles of virus, leading to hemorrhagic fever or severe cases of dengue. Such risk is enhanced. Antibody-dependent enhancement relates to the fact that non-neutralizing antibody combines with the virus, and this leads to the enhancement of the Fc gamma receptor. And the infection enhancement leads to the increased replication of virus, leading to the higher level of viremia, and there is a heightened risk of DHF. To identify this defense mechanism, you first need to identify what kind of immune response could occur through infection or inoculation of vaccine. There may be serotype specific immune response. How this immune response neutralizes the virus, or at what point a neutralization takes place to protect infection. These are the topics of our research. To measure the neutralizing antibody level, we established a new assay using Fc gamma receptor expressing cell. Using this assay, with the existence of antibody, we can measure the viral infection enhancement activity. For reinfected patients, we can also measure ADE activity. Using Fc gamma receptor expressing cell, we carried out and established a new neutralizing test. With this new neutralizing test, we measure the neutralizing titer, and without Fc gamma, the neutralizing antibody was detected, but with Fc gamma receptor, neutralizing antibody was not detected. So this system can measure the antibody activity that reflects the defense level in vivo, and it could be useful in evaluating vaccine efficacy. Using this neutralization test, we looked into viremia level among secondary infected patients. With Fc gamma expressing cell compared to non-Fc gamma expressing cell, the level was tenfold higher, and viremia was sustained for a day longer. Based on these results, the cross-activity antibody without neutralizing a titer combines with virus to form a complex in vivo. This type of assay is useful in reflecting the condition in vivo during reinfection. Using the same Fc gamma receptor expressing cell, we measured the healthy subjects' neutralizing antibody titer and ADE before and after exposure. Among these healthy subjects, neutralizing activity was changed like this. And if you also compare the ADE activity among healthy subjects and patients, among the patients, ADE activity was higher than that of the healthy subjects. So the level of ADE activity is suggested to be useful as defense marker. This new neutralization test using Fc gamma expressing cell, we can measure both neutralizing activity and infection enhancement activity. And we can also measure virus titer for free virus and immune complex. So this system, it can measure neutralizing antibody titer and viral titer. Using the new neutralization test with the Fc gamma receptor expressing cell, this is a useful tool for ADE and neutralizing activity level, and this is mentioned in the guideline of WHO. Neutralization level can be measured with this system, but we also use a new technique, and TCR-BCR gene sequence analysis was tried. Using in silico analysis, we are to predict the gene sequence related to defense mechanism so that we can identify defense mechanism and establish markers for defense and contribute to the antibody-based therapeutics development. In the neutralization test, which is useful for vaccine development, it takes time and labor, so high throughput test needs to be developed. We established the high throughput system, and ELISA-based neutralization test was successfully established. In this test, live virus and strip are used for the high throughput test. Fc gamma receptor expressing BHK and BHK cells can be used for these tests. In the repertoire analysis of B cell, we looked at acute phase 8 months after onset and 1 year after onset of dengue fever. We used PBMC samples for repertoire analysis, and several repertoires were identified in the bulk. Thus, we identified clones, and we also looked at changes of the clones that occur at different stages of the disease using phylogenetic analysis, and we identified several clones successfully. Overall, clone behavior has been looked into, and this is a rapid isolation method of a B cell that we established. Antibody-producing B cell was to be isolated from subjects who are infected. We were able to identify several clones and isolate them. At this point, we are working on the B cell repertoire analysis of the clones. In the meantime, we are also working on the animal model for in vitro study. Another important thing is that we do not have appropriate animal model for the development of the vaccine. Together with the National Institute of Infectious Disease, we are working on the development of a good animal model. Using the model animal, we are to establish the assessment and evaluation method of vaccine and to identify the mechanism of the onset and defense. We use marmoset as animal model for dengue fever. We also would like to use that model to see whether or not it is useful for the identification of the defense mechanism and onset of the disease. Both for secondary and primary infections, the high level of viremia was observed in marmoset, and IgM antibody and IgG antibody levels of the marmoset were similar to those of the human subjects. Because of this, we believe that it's an indication that this is a useful animal model. We also try to see whether or not this animal model is useful for vaccine assessment. We use attenuated dengue vaccine. After inoculation in marmoset, we were able to see the viremia levels. In the challenge study, in the second inoculation, we did not see any viremia level. Similarly, NS1 antigen with the inoculation, there's a detection, but in the challenge study, viral antigen was not observed. So as a result of these tests, we can say that there's a defense established with the inoculation, and the marmoset is useful as an animal model. On the other hand, IgM, IgG antibody induction pattern of marmoset is similar to that of humans. In epidemic regions of dengue fever, it's not just dengue fever, but other flavivirus infections like Zika fever and Japanese encephalitis outbreaks simultaneously occur. In 2015 in Vietnam, there was a major outbreak of Zika fever, and the first case of microencephalopathy was confirmed in Vietnam. According to our epidemiological study, there were small sporadic Zika epidemics taking place from time to time in Vietnam. In 2015, there was a major Zika epidemic, and even before that, after year 2000, there were Asian strains and South American strains prevalent in Vietnam. Zika virus and dengue virus and other flavivirus are prevalent in dengue epidemic regions. And with the flavivirus infection, we need to know what kind of immune response is induced and what interaction these may induce. So using the marmoset model, we looked at dengue and Zika virus infection, and we checked whether there's a cross-reaction in secondary infection. In case of a secondary infection, Zika virus viremia level is higher than that of the primary infection, and for further infection, interferon gamma also was induced. As a result, marmoset shows high level of viremia with dengue and Zika virus inoculation. And in this marmoset model, we identify ecchymosis, rash, and increase of AST and ALT levels. After inoculation, IgM and IgG antibodies were induced, and neutralizing antibody was detected. Based on these results, we can say that marmoset model is useful for dengue virus vaccine and the development assessment of new therapeutics, but there are still many other challenges for vaccine development. For example, there are host-related challenges related to immune response that may take place after infection or inoculation. Fc gamma receptor expressing cell is what we use to measure ADE and neutralizing titer. We also analyze B-cell repertoires. There also are challenges related to vector and virus itself. In case of virus challenges, we need to characterize genes, and antigens have to be studied. For the development of vaccine, we need to establish a useful animal model. We also need to establish a system to develop the assessment and evaluation of the vaccine, including the measurement of the defense markers. Thus, we develop animal model, and using the animal model, we study defense mechanism and disease onset mechanism, and using the accumulated clinical data, we are trying to develop vaccines. These are my acknowledgements. Thank you very much.